everyone, I'm Nathan Hybeck and I'm a bioinformatician here at Zymer Research, and today I have the pleasure of showing you around the Orion Microbiome Database. This is a wonderful platform that will help you with data curation, analysis, and sharing, all related to microbiome data sets. So without further ado, let's jump in. So the first thing you will see when you land on Orion is our login page. Here we have a classic sign-in that you can also utilize Google or GitHub authentication. If you don't yet have an account, you can go here to the Sign Up tab and create an account with your email and password. This will send you a confirmation email with a link to the platform where you can verify your email address, and then you will be ready to go. Let's jump back to the front page and log in. So the first page you will see when you log into Orion is the Workspaces tab. What are workspaces? Workspaces are areas where you can organize your research projects and the collaborators on those projects. As you can see here, I have a ton of different workspaces with a ton of different people. Let's go ahead and create a new workspace here as an example. So we'll create a workspace. Great. Let's name this workspace Demo Workspace and give it a short description. You will also see a summary of all of this over here on the right, and we can also add invitations to collaborators here via their email. Inviting others to your workspace will allow them to see their private data pertaining to that specific workspace, and depending on the role you give them, interact, utilize, and manage that data with varying degrees of freedom. You can also invite users later beyond this workspace creation page and manage their roles independently. We will show all that later on. For now, let's not include any on this round of invites, and let's click Create. And now back to the Workspaces tab. We can see our demo workspace is populated. Let's go ahead and view it. Inside our workspace details page, we can see this top summary tab, which includes some general information like a unique workspace ID, a name, and a description of the workspace. Below, we can also see a list of the members who are currently part of the workspace. Right now, that's just me, and invitations to users that are still pending acceptance. Lastly, we can delete our workspace as well. Now, data is specific to a workspace unless you were to make that data public. Assuming you do not opt to make your data public, that data will remain private to you and the designated workspace that you have ingested it in. That being said, if you would like to collaborate with a few select others within a workspace, you can invite them to join that workspace with the designated role. For example, let's go ahead and add myself to this workspace. Here, I can select a role for myself. These roles dictate the actions and types of access that invited users have. So, a reader can only view the data, while a writer can also contribute to it. Admins have all the abilities of writers, but can also manage user roles and add others to the workspace. And lastly, owners have complete ownership over the entire workspace and reserve the sole power to delete it, or transfer ownership to another user. So, now that we have our workspace all set up, let's go ahead and ingest some data into it. To populate a workspace with data, we will come here to the Submissions tab. Let's make sure that we have the workspace we'd like to bring our data into and is selected here in our top right dropdown. On our submission page, we will be able to see all of our historical submissions for this workspace with a brief summary of each. Right now, we have not submitted anything to this workspace, so it's blank. So let's fix that with a new submission. Every submission is composed of 4 essential parts. You will need a name, a description, a metadata file with your formatted metadata, and your raw sequencing files, which should be in the format of FASTQ or FASTQ.gz. So let's go ahead and give our submission a name and a quick description. Great, now we need a metadata file. For metadata, you are free to bring your own files here, formatted however you would like, but we highly recommend that you utilize our downloadable template and follow the formatting guidelines within it. This template contains a few validation macros that ensure compatibility of your data with our platform, as well as some guidelines for best practices for harmonizing your metadata to be the most compatible with our internal standards. This harmonization is important because it will enable your data to be comparable with large amounts of public data that we have hosted on Orion. If you don't harmonize your metadata, your data will still be usable, but you will lose a large amount of utility from the platform. So, for best results, please download and follow our template, and be sure to enable the macros that come with it. I have already created an example of our template that we will use here, so let's grab that. Great. So I just uploaded my filled out template, and we will move on to the next step, which is harmonization. So here on the harmonization page, you will get a web-based table editor with a few tools that can help you summarize and manipulate your metadata. If you have not used our template, your column names here will be as they were in the file you uploaded, and you may need to manually map them to our internal columns to harmonize. You can do that by going to the More Actions dropdown and selecting the Mapping field. This mapping field, as you see here, will display a list of our standardized internal columns on the right and a list of columns for your metadata on the left. You can manually connect items in this field to make sure your metadata is categorized right while still retaining your original column names. Now, my metadata is already standardized, so all of my columns already exist. But for example, we could map sex to sex, and if we had originally called sex gender in our study, this would be one way to handle that translation. Another example might be what we call host and correlating that to species. So you can go ahead and connect those two, and then we'll see on our table here that we have our new mapping associations, and that will carry on throughout our ingestion process. Again, since we already have our columns harmonized from the template, we don't need these, and I'm going to actually deselect them quickly. Great. Lastly, I want to quickly highlight a few tools that exist on this page that can help you manipulate your metadata or make any last-minute edits. We have a Replace Data tool here to change values in any corresponding columns, and you can also make specific edits to just subsets of your data by applying filters to your columns. For example, we can go to the phenotype and only view the healthy samples, and it looks like we went from our total of 6 samples down to our 3 healthy samples for this data set. Now, while here, we can use our Quick Phenotype tool to additionally tag these samples with a secondary phenotype. Let's say these samples are adults and we want to annotate that. We can apply that phenotype and we will see that additional phenotype populate in rows for just the subset of the samples we have selected. We can then remove our filters to see our full table again. And once we have our metadata squared away, we can click Next to upload our raw data. The final step of submissions will be uploading our raw sequencing files that correlate to our samples. Based on our inputs from our metadata on step 2, we will get a pre-filled out table here that is showing our expected samples, and when we upload our files, they will automatically pair based on our file names from the template. Let's go grab our sample files here and click Upload. Now we see our selected files. Sorry, I cut out the part where I browsed through my local drives, but once we have them, we can go ahead and click Save. Back on our table view, we see that they have already mapped properly to our expected samples, which is great, and we don't appear to have any missing or extra files. If we did, you would see some alerts here in the rows. Now that we are good to go, let's press Submit, and this will automatically trigger those to upload to the portal and run the pipeline. Results from the pipeline will automatically populate once it's complete. Our pipeline does cost to run. It helps cover our cost of compute. And here you will see a final summary detailing how many samples are being submitted and the total credit cost for those samples. So let's click Submit. Great, it looks like we have successfully submitted our run. This takes us back to the submission page where we can now see our new submission. Here is a status bar that will keep us updated on the run status for these samples. And again, upon completion, that data will automatically populate into the data browser where we're headed next. So here on our data browser, you will be able to explore the various types of data we have hosted on Orion, both publicly available data and private data like we just submitted in the last few steps. Now, there is a lot going on here, so let's break it down real quick. Over here on the left, we have some data filters. You can use these to filter the type of data you see here in the browser. If we turn off public data, it will hide all the public files and only show us private data and shared data. Similarly, we could hide our private data and only look at public data by turning off the workspace data selection. Here we turn off public data, but we don't actually have any private data in yet since the pipeline is still running, so you won't see anything populated. That's pretty unhelpful, so let's turn back to public data and look at that. Below that, we have all of our primary metadata categories as well as their associated data point totals for all the samples we are viewing in the browser with our current selection. All these metrics are also graphically represented here within the metrics page, all of which are interactive, allowing you to make selections via the charts or manual filters. Let's go ahead and select some data using the graphs for a quick study. As an example set, we are going to look at colon cancer patients, otherwise known as colorectal neoplasms. Let's select them here in the phenotype chart and we will see it reformat our data and graphs to now show metadata for only those samples. This set looks like it has some pretty diverse metadata, including samples from about 6 different bio projects. Awesome. Next, we can apply an age filter to this data by using the slider tool up here to give us a range of ages selected. Let's go ahead and exclude anyone over 80 for control purposes. Great, it will reformat once again, and now let's also restrict this to only samples from the United States. And then lastly, let's also restrict this to only samples from males. Awesome, this leaves us with a total of 112 samples, down from our about 330 that we started with. Back up top, we can see the series of constraints we put on our data, and we can also view those filters in a more explicit way by opening up the Filters tab at the top of the page. Once here, we can get a more detailed visual of our current filters and the exact patterns they are being executed in, as well as edit, adjust, delete, or add additional filters to our query. In addition to metadata filters, the pre-computed results on Orion also allow us to create queries based on taxonomic profiles. We can set taxonomy-based filters here in the Taxonomic Filters subtab by searching for a phylogenetic rank associated with our organism of interest. For example, let's query for Clostridium difficile and create a search for results related to difficile. Once we search, we will see this result tree populate. This tree identifies the nearest string matches to your search and returns the full lineage of things related to our search. So, we see here we match to Clostridioides difficile at the species level, but also are given matches for that taxa at higher ranks like genus or family. Let's select our species of interest here in the tree that will enable us to create a filter pertaining to an abundance threshold for this organism. We can choose a greater than, less than, or equal to function, and then define the relative abundance percentage we would like to set our threshold at. Now, we don't actually have any interest in this species right now, since we are just interested in colorectal cancer samples. So let's go ahead and delete our query again. Also, back on our primary filters, we are going to remove some of our cutoffs applied earlier to make things a little less stringent. Let's go ahead and remove our age filters, sex, and location, and we'll just keep it as simple as samples that have colorectal cancer. We can save our filter edits and then apply them once again to our data browser. And of course, it will revisualize all of our data again with our new filters, and we are back to our 332 samples that we started with. Wonderful. Now we are ready to take this data onto the next step and begin analyzing it further. Up here at the top of the browser page, you will see two buttons: Add Samples to Cohort and Create Cohort. If we already had a group of samples selected and would simply like to add these samples to that group, we could select Add Samples to Cohort and then select which cohort we would like to add them to. For this set, we want to create a new study, so let's select Create Cohort and create a new cohort for these samples. This will open a pop-up for us where we can enter a title and description for our cohort. Let's name this one colorectal cancer and give it a brief description here. And then let's submit. Okay, so our selection here has been saved as a new cohort that we can now go interact with in the Cohort Studio. Let's go check it out. Here on the Cohort Studio page, we will see a list of all the cohorts we have created within this workspace. Here, we only have one, which is the one we just created, our colorectal cancer cohort with our 332 samples in it. We also have a couple of options along the top to merge cohorts, share cohorts, or go create a new one. Merging cohorts allow you to take two or more individual cohorts you have previously created and combine them into a unique cohort containing all the selected samples. You can also use this function to merge the data from publicly available curated cohorts and merge them with your private data, which is really, really powerful. Those public cohorts can separately be viewed over in the Public Cohorts tab. We can take a quick look, and it looks like right now we just have one hosted here for this demo, but not to worry, many more are coming soon. It looks like we have a mock healthy community from a single NCBI Bio project. How convenient. Let's go back to our Cohort Studio and merge this cohort with our colorectal samples to help compare cancerous patients with a healthy control. To do that, we will go to the Merge Cohorts, and in this pop-up, we will see all of our private and publicly available cohort options to merge. Here, we only have two options, which are the two we want to merge. So let's select them both, then select Merge. We will need to give this new cohort its own name and description. So let's name it Colorectal versus Healthy Cohort and write a brief description. Great, and now we will see our new cohort in our Cohort Studio with about 430 total samples. Let's jump into it and start to explore. When viewing a cohort within the Cohort Studio, you will be met with a very familiar metrics page that is the same page you used in the data browser to create this cohort. This is so you will be able to do further refinement of your cohort later by adding more filters. So for example, let's say that we finally decided to put an age filter on this cohort, and we can apply one here. So let's go with 68 as our age cutoff and apply that filter. Just as before, we renormalize our data, and now let's push this edit to become a permanent restriction on this cohort by selecting the Edit Cohort button. We will get a log here of our changes being made where we can add some additional notes, and it also captures the filter changes that are being added to this cohort. We don't want to keep this change, but if we did, we would just have to select Submit and our new cohort would be modified. So back on the metrics page, again, you will have the full utility of these graphs and filters. Then additionally, there is a table view of your cohort samples where you'll be able to explore your data in a more spreadsheet style. Let's change our rows here to 100 and then look through some of this. A few things you will be able to identify here is the workspace origin for these samples. And as you can see here, these samples are from the public workspace, so they must be from our public cohort that we merged with. We also have access to our filter functions here and can apply them to the table view as well as pre-existing filters created on the metrics page. So if we have no further edits for our cohort parameters, then we can continue to the Plots tab, which is where we will be able to visualize our current data selections with the pre-computed results hosted on Orion. When you go to the Plots tab, you will first find a three-dimensional PCOA chart of your samples under the Clusters subtab. This chart can help you identify broad themes within your dataset, especially in relation to metadata categories. If we look here under the Columns dropdown, we see a selectable list of all of our metadata fields that we can choose to color our samples by on the chart. Right now, it's colored by sex, but let's go ahead and change the categorization to health status. And when we do that, we see we have a pretty significant separation of two distinct groups. We can also visualize this based on other metadata fields such as location or sequencing platform to help identify potential outliers that might stem from different methods in sample collection, regional diet, or analysis methods. We can also visualize some continuous variables here like age, which will alter our dots to exist on a gradient. Let's jump back to our health status view here and select a few samples to explore further. We can select a few points within our graph, and as you see, they will become highlighted, and then we can head over to the Composition subtab to look at the taxonomy profiles for these two samples. On this page, we will get a pretty standard composition bar plot of our relative abundances for each sample at the species level by default. If we hover over the bars, we will see the species name and relative abundance displayed in a small pop-up box. We can also plot just specific taxa by using the Plot Taxa tool over here on the left. This drop-down will display all the available results in our samples that we can select from as the search bar along the top. Let's select a few organisms here to view in the plot, and you will see that when we select taxa here, it changes our plot to only display the relative abundances of what we have selected. This way, we can directly compare the abundance differences for any organism of interest between a set of samples and explore how those distributions change. Okay, let's clear this setting now and go back to our original view, where we can also revisualize our relative abundances at a different level of taxonomy using taxa level dropdowns. This dropdown will allow us to select any level of taxonomy to plot our abundances for. We are going to skip this step for now and instead are going to actually remove a sample from our current cohort. To do that, we can go to our Selected Samples button and deselect one of our samples so that we only have one selected. And then let's go to the Edit Cohort button. In this pop-up, we will get a space for change notes related to this edit and an option at the bottom to either take a subset of our current cohort with our current selection or remove the currently selected points. Let's elect to remove this sample and say it has an error in it, and then press Submit. After our cohort edit takes place, our cohort visuals will automatically regenerate and we will be able to track this edit in the History tab up on the top right. Here, we see a version control of our cohort along with the edits we have made to it, including days, times, users, comments, and most importantly, a revert tool that will allow us to safely revert back to a previous version of a cohort in case we made a mistake with one of our edits. Let's do that for our last edit to restore our cohort back to its former glory, and then we will also see that revision populate in the History tab as well. That is currently all we have in our Cohort Studio in terms of functional tools, but we are actively working on bringing some more utility here, including LeftC, a differential abundance analysis tool that will be going live very soon following launch. as well as many more analysis features in the future, such as comparative analysis, modeling tools, AMR analysis, and more. Well, that summarizes all of our current utility on the Orion platform and user workflows through each of our steps. Thank you so much for viewing this tutorial and participating with the effort that is the Orion project. We love community engagement and ideas on ways that we can continue to improve this platform to best serve the research community. So if you have any questions, thoughts, or feedback, please reach out to us through our support email, which can be found within the Contact Us tab under the user profile dropdown. Once again, thanks for tuning in, and we here at Orion Enzyme Research wish you all the best in your adventures through the microbial universe. Toodaloo.